So here this is illustrated again in terms of the development of the deflection point, the point at which the cell wall opens to allow the transmission of neurotransmitters, and the period leading up to this change is called the absolute refractory period, and the period after is called the relative refractory period. So there is a rebalance back to the normal state. So now I'd like to turn to discuss the differences in structure in the neuronal system and what that tells us about some types of neuropathology. Let's begin with the dendrites and how a typical or normal dendrite would look compared to an abnormal dendrite that we sometimes see in certain cases of neuropathology. So the normal dendrite will always have very similar shaped structure, so that we may see very little variability in the length of the spines and also the location of the different oligodendrocytes as well. But in the case of an abnormal dendrite, we will see a kind of frazzling of these spines that look irregular in shape, they seem ill-formed, and they are a little bit like unbrushed hair. In the case of children with learning difficulties, say Down syndrome, what we see is a difference between the dendritic structure of the brain cells in the case of Down syndrome compared to people or children who do not have learning disabilities. So we know that by looking at through the microscope of the brain structure of children with Down syndrome, they have very particular dendritic shape. It's important to realize that doesn't mean that the dendrites do not work. They do work. It's just that the structural properties of the dendrites are very different. If we take a look at the changes in neuronal structure that occur through pathology such as cancer, then we can also see some specific types of metastatic changes to the typical brain cell structure. So a primary brain tumor would be seen as a mass or a lump inside the cortex. Now, this is no different to any other organ of the body. So the brain depicted here with the left and right hemispheres could be a liver, it could be a kidney, it could be any organ. Within the healthy tissue of the organ, a tumor can develop through a number of different reasons, but the tumor itself simply describes an abnormal growth of cells, particularly an abnormal reproduction of cells at an extraordinarily fast rate, which leads to a mass that creates pressure on the brain and also interrupts the normal function of the brain and can also create some life-threatening illnesses, which will be the secondary outcome of the tumor itself. The tumor is formed in a number of different ways. It depends upon the type of cell which is showing irregularity. So the cells that make up the nervous system that we have reviewed, all those different forms of cells, they can all become cancerous. So one can have a Schwann cell melanoma, for example. One could have a cancer that is driven by changes in the subcortical pyramidal neurons, which are depicted here. One could have a cerebella. So the point about the brain tumor is that it represents an abnormal proliferation of cells, any one of the cell types that we have illustrated so far. But to be able to remedy the brain tumor, we need to know what type of cell is causing the tumor so that proper medication can be administered. Here's a scan of a brain tumor, and it illustrates the use of PET technology, positron emission technology. So recall that positron emission tomography is able to pick up metabolic changes in the brain and can illustrate these metabolic changes in real time. And the way that's done is by looking at the activity of glucose inside the brain, the working brain. A high level of glucose activity illustrates that there's more activation taking place. So the red areas here illustrate much more glucose uptake. The yellow areas sort of middling amount of uptake, as you would expect in a typical awake brain. And the green and blue areas represent much less activity. So there's very little uptake of glucose in those areas that are blue. What you can see here is that there's a couple of identifiable features. There's the ventricle, but there's a very large space right here, which is blue, and suggests that there is no metabolism taking place in this area. So that would suggest that either the area is no longer alive because it's not metabolizing glucose as it should do if it was still living cells. But if the cells are no longer living, then what we should see is a relatively low amount of glucose metabolism. And that's exactly what we see. So this suggests that there is a brain tumor, particularly because the locus of the blue patch is fairly symmetric in its round shape. It's on one side of the brain, so we only see it on one half and not the other. And it looks suspiciously to the radiographer and the neurologist like a space-invading lesion, which is essentially dead tissue.